Welcome back to the Deep Dive. We're here to give you the context behind the science making headlines.
And today, we're diving into single-cell biology.
Yeah, it's a field that's just exploded. We get these incredible technologies that give us, billions of data points per experiment. Unbelievable detail on cellular differences, heterogeneity.
It really is amazing, but, the sheer volume isn't the main hurdle anymore.
Right. It's making sense of it all, isn't it? The complexity. How do you reliably link these sometimes really subtle changes in cell populations, the phenotypes, to the things that actually matter clinically, like, disease status or how severe it is?
That's precisely the issue. Many of the big biological questions, the ones with clinical impact, they aren't simple yes-no questions about whether a cell type is there or not. It's more about conditional effects, like maybe a certain immune cell population only drives severe disease if the patient is, say, female or over a certain age.
And traditional methods struggle with that.
They really do. A lot of the standard tools rely heavily on linear models. They're great for finding straightforward associations, but they often fall short when the biology is driven by these complex, multiple factors interacting. They just aren't designed to pick up on those nonlinear interaction effects.
Okay, so that's the gap. If your analysis tool can't see that hidden connection like disease status interacting with sex or age, you could be missing the most important part of the story.
Entirely. You might miss the key driver.
Which brings us to today's focus. CellPhenoX. You've been looking into this.
I have. It's an explainable machine learning method, the X is for explainable, and it's built specifically for this problem.
Okay.
It digs into single-cell multi-omics data, so transcriptomics, proteomics, to find those specific cell phenotypes that are influenced by these complex interactions, and then it links them to clinical outcomes.
And explainable part is key here, I imagine.
Absolutely. Our mission today is really to unpack how CellPhenoX uses principles from explainable AI, or XAI. It moves beyond just saying there's a statistical difference to generating a score.
An interpretable score.
Exactly. A robust, interpretable score for each cell that tells researchers pretty clearly which cells are actually pushing the model towards classifying a particular disease outcome. It translates that complexity into a signal you can potentially act on.
All right, let's set the stage a bit. Before tools like CellPhenoX came along, how were researchers trying to figure out which cell populations were changing in disease? What were the standard approaches?
Well, the field evolved, Initially, a lot of it was based on clustering cells into discrete types and then testing if the abundance of those clusters changed between conditions, like more T cells in disease versus healthy.
Okay, classic clustering. But that can be a bit rigid, right? Biology isn't always neat boxes.
Exactly. So methods evolved towards cluster-free approaches. Tools like Milor and CNA became quite popular. They look at cells within their transcriptional neighborhoods.
So more granular. Less about forcing cells into predefined boxes.
Right. It allows for detecting shifts in more continuous cell states or smaller subpopulations. And they had advantages. Milor, for instance, was noted for being pretty accurate even when you had batch effects, which are always a headache in these big experiments.
Yeah, batch effects are the bane of bioinformatics. And CNA.
CNA was highlighted for its scalability. It could handle larger data sets quite well, so definite progress. These methods were, and still are, very useful.
But they still had limitations, which led to the need for something like CellPhenoX.
Yes. And you actually hit on the core issue earlier. Their statistical foundation is almost entirely built on linear mixed effects models.
Which are powerful for certain things.
Very powerful for testing direct statistical associations, yes. Is this neighborhood significantly more abundant in condition A versus condition B, adjusting for confounders? They do that well.
A fatal flaw.
Yeah.
What couldn't they do effectively?
Two main things, really. First, they aren't inherently predictive in a machine learning sense. They test associations. They don't build a model optimized to predict the outcome. But the bigger issue, the one CellPhenoX directly tackles, is that they are fundamentally not equipped to reliably detect cell populations whose abundance changes are driven by those complex interaction effects.
Okay, spell that out a bit more. The interaction effects.
Think about it like this. A linear model can tell you, okay, having the disease is associated with a change, and maybe being female is associated with a change.
Right, two separate effects.
But it struggles to quantify the scenario where, say, a specific subset of regulatory T cells dramatically expands only if the patient has the disease and is female. It's not additive, it's multiplicative, conditional. The standard linear models used in Malor or CNA just don't have a straightforward way to test for that specific sex times disease interaction within a defined cellular neighborhood or population.
I see. So you might find the main effects of disease and sex, but you completely miss the crucial interplay between them that defines a specific patient subgroup's response.
Precisely. And that interplay might be the most biologically or clinically relevant finding.
Which is where AI, specifically explainable AI, comes into the picture. We need something that can handle that nonlinearity, that complexity, especially with messy, real-world data, multiple samples, different disease stages, demographic factors, all mixed together.
Exactly. The shift to AI isn't just for predictive power, it's for uncovering these hidden patterns. And explainable AI, XAI, is critical because we can't just have a black box predictor. In biology, in medicine, you need to understand why the model is making a certain prediction.
You need to trust it and understand the mechanism.
Absolutely. So CellPhenoX leverages a specific XAI technique called Shapley additive explanations, or SHAP values.
Yes, SHAP values. Okay. Where do they come from? What's the idea?
They're actually rooted in cooperative game theory. Imagine you have a team of players who score some points together. How do you fairly distribute the credit for the final score among the players? SHAP does something similar for machine learning models.
Okay, interesting analogy. So the players are the features going into the model, like the cell abundance dimensions, the patient's age, sex.
Exactly. And the final score is the model's prediction, like probability of having the disease. SHAP calculates the marginal contribution of each feature to that prediction for every single data point, in our case, for every single cell.
And how does that help with the nonlinearity, the interaction problem that stumped the linear models?
Because SHAP considers all possible combinations, all possible coalitions of features when calculating these contributions, it doesn't just look at a feature in isolation. It assesses its impact in the context of all other features.
Oh, so it inherently accounts for how features work together.
Precisely. It can quantify that synergistic or antagonistic effect when multiple factors combine. It gives you a local importance score. How much did this feature push the prediction up or down for this specific cell, considering everything else?
So SHAP values finally let us ask that question. How much did being this type of cell from a female patient with high expression of marker X contribute together to the prediction of severe disease?
That's exactly the kind of question it enables you to answer quantitatively. And that transparency, that ability to assign contribution, even in the face of interactions, is the core engine driving the CellPhenoX interpretable score.
Okay, let's get into the nuts and bolts. How does CellPhenoX actually work? You said it integrates classification robustness with XAI transparency. What are the steps?
Right. It's a multi-step process. Step one is really about data preparation, creating the features the model will learn from.
Makes sense. Garbage in, garbage out,
Yeah.
right? So what's the input? Raw single-cell data.
Yes, typically single-cell gene expression or protein data. The first thing CellPhenoX does is transform this into what's called the neighborhood abundance matrix, or NM.
NAM. Okay, unpack that for us. What is a neighborhood abundance matrix?
Instead of just looking at individual cells in isolation, it tries to capture the local context. It builds a nearest neighbor graph based on cell similarity, usually in the gene or protein expression space.
So finding cells that are close to each other biologically.
Exactly. Then for each cell, it defines a neighborhood on this graph. The NAM then quantifies how abundant or dense that specific type of neighborhood is across all the different samples in your study.
Ah, I see. So it's not just cell A has this gene profile. It's more like in sample 1, neighborhoods like cell A's neighborhood are very common, and maybe in sample 2, they're rare.
That's a great way to put it. It shifts the focus from single cells to the relative abundance of these local cellular microenvironments or states across samples.
Okay, so now we have this NAM. Sounds like it could be pretty high dimensional, right? Lots of neighborhoods.
It usually is, which is why the next step is dimensionality reduction. They need to boil down this NM into a smaller set of key patterns or latent dimensions. These latent dimensions, let's call them $6, become the actual features fed into the classification model.
And I notice they offer choices here. Principal component analysis, PCA, or non-negative matrix factorization, NMF. Why both?
That's about flexibility, adapting to the data. PCA is sort of the default workhorse. It finds orthogonal dimensions, principal components that capture the maximum variance in the NA. It's great for finding the most dominant general patterns of variation in cell abundance.
Okay, standard PCA. What about NMF? Why use that?
NMF comes from fields like topic modeling. The key difference is that it forces the resulting dimensions or factors and their contributions to be non-negative. This constraint often leads to factors that represent more distinct, interpretable biological programs or topics within the data.
So NMF might give you dimensions that map more cleanly onto, say, an inflammatory response neighborhood versus a tissue-resident neighborhood, more intuitive parts-based representation.
Potentially, yes. It can sometimes provide features that are easier to interpret biologically compared to PCA components, which can be combinations of many things. So having both options lets researchers choose based on their data and goals.
Smart. Okay, so we have our features, the latent dimensions, $6 from either PCA or NMF. Now, step 2, training the classification model. The goal is to predict the clinical outcome, dollars, using these features, $6. But you said CellPhenoX handles complexity differently here.
Yes, this is crucial. When they train the classifier, they use random forest, which works well with SHAP. They don't just use the NAM dimension $6, they explicitly include other variables as predictors in the model. Like known technical covariates, maybe the sequencing match, and critically, those biological variables you suspect might interact with the cell states, like age or sex. And any interaction terms you hypothesize are important, like age times $6 or sex times $6.
Wait, so they don't regress out batch effects or age beforehand, they put them in the model.
Exactly. They don't remove that information. By including covariates and interaction terms directly in the random forest model, the model learns their effects and how they potentially modify the effect of the cell abundance features. This preserves the full context.
Why is that important? Why not just clean the data first?
Because if you regress out age first, you lose the ability to specifically assess how cell state abundance interacts with age to predict the outcome. By keeping everything in the model, the subsequent SHAP analysis can tell you precisely how much the age times $6 interaction term contributed to the prediction for each cell. You maintain interpretability of those interactions.
That makes sense. You need the interacting factors present to quantify their interaction.
Okay, and they use nested cross-validation, CV, for training. Sounds robust. Why nested? Quick explanation.
Yeah, nested CV is important for rigorous model evaluation, especially when you're also tuning model parameters, which random forest has. Standard CV might give you an overly optimistic view of performance.
Because you might tune the parameters on the same data folds you use for testing performance. Nested CV uses an outer loop to split data for final testing, and an inner loop within the training portion to do the parameter tuning. It strictly separates tuning from final evaluation.
Got it. So it prevents the model from peaking at the test data during tuning, giving a more honest estimate of how well it will perform on completely new, unseen data. Prevents overfitting.
Precisely. It ensures the performance estimate is unbiased and the model is likely to generalize.
All right, robust training. That leads us to the final step, the real payoff. Step 3, calculating the interpretable score.
This is where the XAI comes in full force. After the robust random forest model is trained, they apply SHAP. SHAP calculates those contribution values for every feature the NAM dimensions $6, the covariates like age, sex, batch, and those crucial interaction terms for every single cell.
Okay, so you get a bunch of SAP values per cell, one for each input feature.
Exactly. Then, to get the final interpretable score for a cell, they simply sum up all the SHAP values associated with that cell across all the features.
So the score is a single number per cell that summarizes its total impact on the model's prediction.
Yes. It represents the cell's overall contribution towards the predicted clinical outcome, considering all main effects and interactions. If a cell has characteristics strongly pushing the model towards predicting disease, it will have a high positive score. If it pushes towards healthy, maybe a high negative score. If it's irrelevant to the classification, its score will be near 0.
And this is the score you can visualize, like color-coding cells on a UMP plot.
Exactly. You project the specimen score onto your UMP or similar visualization. Suddenly, you can visually see which groups of cells, which phenotypes, are getting high scores, meaning they're the ones driving the classification. It directly links the prediction back to the biology you can see.
Very cool. What if you have more than 2 outcomes? Like the COVID example, healthy, mild, moderate. How does the score work then?
Good question. In multi-class scenarios, SHAP provides values for each possible outcome class. CellPhenoX handles this by aggregating the SHAP values based on the class that the model predicted for that cell. So each cell still gets one single interpretable score, reflecting its contribution toward its predicted outcome class. It keeps the interpretation focused.
Right. Theory sounds good. Mechanism seems clever. But does it work in practice? Let's talk about the validation. They tested CellPhenoX against Malore and CNA using simulated data first, right?
They did. Simulations are great because you know the ground truth. You design the data set with specific changes and see if the method can find them.
So what were the key findings from these benchmarks?
One really stood out. CellPhenoX showed superior ability to detect rare cell populations that were changing.
Rare cells are often really important biologically, but easy to miss.
Exactly. In one simulation, they designed a rare cell type called cluster J to be expanded in the disease condition. CellPhenoX's interpretable score clearly highlighted this cluster. Milor, on the other hand, completely failed to identify it.
Wow, okay. So Milor, despite being cluster-free, maybe missed that subtle signal from a low abundance population.
That seems to be the case in this simulation. It suggests CellPhenoX might be more sensitive to these potentially critical rare cell shifts while still being robust.
That's a significant advantage. What else?
They also tested a scenario with very subtle changes, simulation dataset 2, where the fold change in abundance was only 0.1, very small.
Harder to detect.
Much harder. And interestingly, they found that the CellPhenoX interpretable score was actually better at separating the disease-associated cells, clusters A and J, in the sim, than just using the raw prediction probability from the random forest model itself.
So the SHAP Bates score adds discriminatory power. It's not just for interpretation, it actually refines the signal.
That's what the results suggest. The score, by summing up the contributions, seems to provide a clearer signal of biological relevance than the raw probability alone, especially when the underlying effects are weak.
Okay, better on rare cells, better on subtle effects. But the real unique selling point was handling interactions, right? The thing linear models couldn't do. How did they test that?
They designed a specific simulation for that. They created a scenario where a cell population shift, again, let's say the expansion of that rare cluster J, was dependent on an interaction between disease status and sex. It only happened in female disease samples.
The exact kind of conditional effect we talked about.
Precisely. They ran CellPhenoX, including disease status, sex, and the PC1 times sexin interaction term as features, along with the AM principal component PC1.
And what did the SHAP analysis reveal? Which feature was most important?
The most salient feature, the one with the highest overall SHAP importance for driving the classification, wasn't PC1 alone, and it wasn't sex alone. It was the interaction term between PC1 and sex.
The model correctly identified the interaction as the key driver.
Yep.
Okay, now connect that back to the interpretable scores for individual cells. How did they look?
It was really clear. For cells from the simulated male individuals, both healthy and disease, the interpretable scores were all hovering around 0.
Meaning those cells weren't contributing much to the disease classification in males, according to the model.
Right. But for cells from the simulated female individuals, there was a dramatic difference. Cells belonging to that expanded cluster J in the female disease group had strongly positive interpretable scores, around plus 0.35. And cells from depleted types in that group had strongly negative scores, maybe negative 0.35.
That's incredibly specific. A quantitative readout showing the cellular change is strongly associated with disease, but only in females.
Exactly. It provides that clear quantitative signal of a sex-biased cellular mechanism that traditional differential abundance tests relying on linear models would likely miss or struggle to quantify properly. This is the kind of nuanced insight researchers need.
Yeah, you can imagine clinicians observing patterns like this, but having the statistical tool to actually pinpoint the cellular basis and the interaction driving it, it's powerful. Okay, simulations are one thing, but the real test is on messy, complex patient data. Let's look at the first case study. COVID-19 severity using single-cell proteomics. This sounds challenging.
Definitely. It was a multi-class problem, classifying patients into healthy, mild COVID or moderate COVID stages. And the data was, as you say, messy.
What kind of messiness?
A lot of potential confounding factors. They had to adjust the model for multiple covariates: patient age, sex, the number of days since symptom onset, smoking status.
Okay, standard clinical variables.
Plus interaction terms for age and sex, and they used Harmony for batch correction across different experimental runs. So a really complex setup trying to mirror a realistic clinical cohort.
Right, a tough test. So after running CellPhenoX through all that, what emerged? Which cells were pointing towards moderate severity?
The strongest signal came from the myeloid cell population. That's perhaps not surprising, given the role of myeloid cells in COVID inflammation.
But did it get more specific than just myeloid cells?
Yes, it did. Within the broader myeloid group, the interpretable scores particularly highlighted a specific, relatively small subset: proliferating monocytes. These showed a distinct, strong signal linked specifically to the moderate disease stage.
Proliferating monocytes. Okay, a minor but potentially crucial phenotype driving that moderate severity classification. And what about the features? Which feature or interaction was the most important predictor overall in the model?
This was really interesting. The dominant feature, the one contributing most to the model's predictions across the classes, wasn't just a cell abundance dimension nor age alone. It was the interaction between the first principal component of the NAMPC1 and patient age.
Wow, hold on. So the way the overall myeloid cell landscape captured by NMPC1 relates to COVID severity is significantly modified by the patient's age.
That's what the model is telling us. The contribution of those cell abundance patterns to predicting severity depends heavily on age.
That aligns with so much clinical observation, doesn't it? We know age is a huge risk factor, and the immune response differs dramatically, but CellPhenoX seems to quantify that interaction down to the cellular neighborhood level.
Exactly. It's providing a potential cellular mechanism linked to myeloid cells and specifically proliferating monocytes for why age is such a critical factor in severity, driven by that quantifiable interaction effect.
Did they validate this biologically? Did the scores correlate with anything known?
They did. They found that the interpretable scores correlated significantly with the expression levels of known COVID severity markers, often found on myeloid cells, like Siglec-C1 and CD33.
Okay, good sanity check.
And also with key immune complement components like C5AR1 and CR1, which are involved in the kind of systemic myeloid activation you see in more severe inflammatory states. So the score tracked with relevant biology.
Very compelling. Okay, let's switch gears to the second case study, ulcerative colitis, or UC. Here they used single-cell transcriptomics from gut biopsies, right? And focused on fibroblasts.
That's right. Fibroblasts are increasingly recognized as key players in gut inflammation and fibrosis in UC. The goal here was simpler. Predict inflamed versus non-inflamed tissue status using only the fibroblast data.
And they used NMF for dimensionality reduction this time, looking for those biological topics.
Yes, they opted for NMF here, and perhaps unsurprisingly, the analysis showed that the cluster identified as inflammatory fibroblasts had the highest average interpretable score in the inflamed samples.
Confirms their role, okay.
Yeah.
But did the score reveal anything beyond just this cluster is important? Any finer detail?
Yes, this is where the visualization really helped. When they plotted the interpretable score on the UM8, it didn't just show two separate blobs, non-inflamed and inflamed fibroblasts. It revealed something more like a continuous transition, a gradient.
A gradient,
Mhm.
meaning
Meaning the score suggested a progression. It looked like cells were transitioning from a state characterized by markers like WNT2B plus dollars, which was associated with non-inflamed tissue and had low scores, towards the inflammatory fibroblast state, which had the highest scores.
so it's not just two distinct types, but potentially a developmental path or activation trajectory towards inflammation, and the interpretable score maps out that path.
That's exactly what it suggested. The axis of that transition, captured by the gradient in the score, seemed to be a key predictor of the tissue's inflammatory status.
That offers a much richer biological picture. Maybe interventions could target the transition itself, not just the end state.
Potentially, yes. And again, they've validated this with gene correlations. Genes known to be involved in inflammation, like CCL90 signers and col6A33, were positively correlated with the score along that gradient.
Higher score, more inflammatory markers.
Right, and genes characteristic of that starting WNT2B plus DOSR, like ADAM-DEX21 and CXCL12-2, were negatively correlated. Lower score, more of the origin state markers.
So the score accurately captured the molecular changes happening along that potential transition axis.
It really seems to map that biological process driving tissue status quite effectively. #DatTrack.
Okay, this has been a really insightful deep dive into CellPhenoX. Let's try to summarize the core value here. What makes it stand out?
I think the unique proposition is that combination. It delivers cell-specific interpretable scores, which is huge for biological understanding. It handles complex data, multi-omics, multiple clinical groups.
Yeah.
But critically, it's designed to capture and quantify those interaction effects, how factors like age or sex modify cellular contributions to disease, which, as we've discussed, the more traditional linear methods often struggle with or miss entirely.
It finds the conditional patterns. Now, no tool is perfect. Did the authors acknowledge limitations?
They did, yes. One practical limitation is computational cost. Calculating SHG p-values, especially considering all feature interactions, can become very computationally intensive, potentially limiting its use on truly massive, like million-cell plus datasets without significant resources.
Okay, scalability could be a challenge for the biggest datasets. Anything else?
They also mentioned that its power might be somewhat reduced in situations with extreme confounding. For example, if all your young patients were processed in batch 1 and all your old patients in batch 2 and maybe came from different tissues.
Oh, where the factors you want to separate are almost perfectly overlapping.
Exactly. If covariates are highly collinear or confounded, it becomes statistically very difficult for any model to perfectly disentangle their individual and interactive effects. CellPhenoX includes them, but extreme confounding can still dilute the signal.
Fair enough. Fundamental statistical challenges remain. But assuming reasonable data quality, the potential seems huge.
I think so. The framework itself seems quite generalizable. They focused on transcriptomics and proteomics, but there's no reason it couldn't be adapted for other single-cell modalities, like ATAC-seq for chromatin accessibility, or even spatial transcriptomics data.
Integrating location as a feature or covariate. Interesting.
And the implications for translational research are pretty clear. Being able to predict drug response based on specific interacting cell states, track disease progression more granularly, or finally get a quantitative handle on those sex or age-specific cellular mechanisms, it's powerful.
It really bridges that gap between complex data and interpretable, potentially actionable biological insight.
That seems to be the goal.
So let me throw a final question out there, both to you and to our listeners. Since CellPhenoX gives us this ability to pinpoint how much individual cells, defined by complex features and interactions, contribute to a disease outcome, how do you see this changing therapeutic targeting in, say, the next 5 years? Are we going to move beyond just targeting T cells or fibroblasts broadly and start developing drugs aimed at very specific cellular phenotypes that are only relevant in certain patient subgroups defined by these interactions, like proliferating monocytes in older males or WNT2B plus transitioning fibroblasts in females?
That's the dream of precision medicine, isn't it? If, with this level of interpretable detail, exactly which rare cell subset, perhaps only active under specific conditions like age or sex, is truly driving the pathology, then yes, your therapeutic target becomes incredibly refined.
From sledgehammer to scalpel.
Potentially. That shift from population averages, which might mask the critical subgroup, to understanding these specific context-dependent cellular contributions. I agree, that feels like the path forward. It could fundamentally change how we think about biomarkers and drug development.
A very compelling future to consider. Thank you so much for walking us through CellPhenoX today.
My pleasure, this fascinating work.
And thank you for joining us on the deep dive. We'll catch you next time.